In this video, we're going to be talking about a topic that we'll cover in a lot more detail in organic chemistry too. It's called resonance structures. And so just getting right into it, resonance structures are hugely important. They're very interesting, and they allow us to understand reactivity in a lot more detail. I used to teach them in organic chemistry 1 quite a bit, but I found that students didn't really need them except for the Grignard reaction until organic chemistry 2. And as a result, I just kind of decided to wait until then to talk about them in a lot more detail. So some of this might seem a little awkward, but it's actually quite useful for understanding organic chemistry, in particular the mechanistic aspects. So getting into it, resonance structures are just different ways of drawing the same molecule. So how do these different drawings differ if the molecule is the same? The drawings differ in the distribution of electrons, in particular pi electrons. Okay, that is to say, if I just get right into it, the pi electrons between the carbon-oxygen double bond of acetone actually aren't fixed in position. They kind of, they are a density, a cloud. They can kind of flutter back and forth between the two atoms, and in particular, they prefer to be around the more electronegative atom, the oxygen atom. And so much so that one could draw acetone like this. So these two structures are actually the same. They're both acetone. What we've done is just drawn them differently. They're not isomers of each other. They're different depictions. This is an alternative depiction. An alternative depiction, which is a resonance structure. In addition to the structure, I've also included the resonance arrow, which is a double-headed arrow, different from an equilibrium arrow. It's a double-headed arrow where the heads are on both sides. And this just shows and communicates to us that the electrons between the carbon and oxygen atom in one depiction, the first depiction, could actually be considered a lone pair on the oxygen atom. Both are effective depictions. So acetone, the 2 pi electrons could be, number 1, between the 2 atoms and a double bond, or sort of giving a double bond. Alternatively, they could be on the oxygen as a lone pair. If we put them on oxygen as a lone pair, then C is positive and O is negative. You can do this with really any double bond. So if we have a carbon-carbon double bond, we could, instead of having 2 lines between the carbon atoms, we could put an extra lone pair on one of the carbon atoms. It would then be negatively charged, and then a positive charge on the opposite carbon atom. We'll do this a lot more in organic chemistry too and show why it's useful. For now, it's useful because it allows us to see in more clearly why the Grignard reagent prefers to add its anion-like carbon to the carbonyl carbon as opposed to the oxygen atom. The last thing I'll say about resonance structures is that resonance structures can interconvert using curly arrows. So our curly arrows, these are the things that allow us to see how electrons move during the course of chemical reactions. Here we're showing how electrons move and get redistributed across molecules. Now, no nuclei change position, which is unlike a chemical reaction where we kind of have a nucleophile attacking an electrophile. All of the atoms and their respective nuclei go along for the ride, but the nuclei don't change position when we're drawing resonance structures. It's the electrons that change position, I guess. It's actually the electrons are where they are, but we can't just draw them with lines and circles and that sort of thing and get the full picture of what's going on inside the molecule. I really like to say that resonance structures make up for a shortcoming of bond line structures. Okay, so what I've shown here is a curly arrow that shows the electrons moving from the double bond onto the oxygen atom, and I give it a lone pair. I should also, a negative charge. I should add the lone pairs of electrons so we have a lot of clarity here. But now, after the electrons move, the formal charge on the oxygen atom is now -1, because we have group 6 elements minus 6 lone pairs of electrons minus 1 bond is equal to -1. Whereas the formal charge on our carbon atom is a group 4 element minus 3 bonds minus 0 electrons, that's +1. Okay, so we've accounted for the charges being present, and we've used an arrow, one of our just traditional curly arrows, to show how the electrons are redistributed in the acetone molecule. What I want us to pay attention to is that during the mechanism for Grignard reagent addition, what we could do is we could have one of two things occur. Let's take our R- from the Grignard reagent. We talked about this in the last lecture. So from the Grignard reagent, and we could add that to the carbonyl carbon, and we could just know that carbonyl carbons get attacked by the carbanionic Grignard reagent. Now, when we add to that carbon, we need to push the electrons up onto the oxygen atom, otherwise the carbon will exceed its octet. When we do that, we get to something like this. And this is our adduct, which is then quenched in the third stage of the Grignard reaction. Alternatively, what we could do is we could have our carbonyl, our acetone molecule, which is provided in this structural form. We could just redraw it as a resonance structure. It's a different depiction of the same molecule. If I ask the question, is this structure acetone or is this structure acetone? The answer is yes, an awkward answer. But it just tells you that acetone is both of these molecules, okay? So both of these molecules are acetone. Now the advantage of drawing the resonance structure is it's more clear where the negative charge is going to attack. If it has a choice, it's probably going to attack the atom with a positive charge. Opposites attract, we neutralize the positive charge, and we actually get our oxygen atom with a negative charge because it had a negative charge as its resonance structure. So we only need one curly arrow to show the addition of the anionic carbon from our Grignard reagent to the carbonyl carbon, and then we're done. Okay, we get to the exact same outcome. Resonance structures are different depictions of the exact same molecule. They do the exact same things as the other molecule. You're not going to open up new chemistry, but it's a little bit easier to see how things might behave. So I can't emphasize this enough. The same molecule. Acetone's just drawn two different ways. You can draw double bonds conveniently as two lines, or with a charge separated system, putting a plus charge on one atom and a minus charge in the other. Could we have flipped them around? Could we have put the positive charge in the oxygen, the negative charge in the carbon? Technically, yes, although it doesn't behave like that. And so it's not a useful resonance structure. We'll talk about that in organic chemistry too. It's not useful because oxygen is so electronegative. So what I want us to do is just see why I was drawing the charge separated species. What this is really taking advantage of is it's allowing us to, it's advantageous because it allows us to see how things are going to kind of combine together, especially when charges neutralize each other. Another thing to keep in mind, we've been drawing a lot of molecules in two-dimensional pieces of paper or iPad screens, whatever. We're not going to get it right all the time. And resonance structures account for times where we don't get it right. That is correctly portraying the distribution of electrons in pi bonds. We don't get right by just drawing double bonds unless the two atoms are the same. Resonance structures allow us to account for this and take kind of come up with some way of correcting for a shortcoming of bond line structures. Having a bunch of resonance structures is actually a better way of depicting a single molecule.